What if I told you one of the most powerful opportunities to repair your brain happens every night while you sleep? It does not occur in a doctor's office, does not require a prescription, and does not cost hundreds of dollars in supplements. What you eat in the 2 hours before bedtime can either speed up brain aging or actively reverse it. Many over 60 believe memory loss, brain fog, and declining cognition are simply unavoidable consequences of getting older, something to accept and manage. Emerging research shows this is not true. The aging brain is not broken. It is simply deprived of the right signals, nutrients, and environment to repair itself. The window right before sleep, when the brain performs its most critical housekeeping, is one of the most underused opportunities in health. 3 specific fruits, easily accessible and scientifically validated, can provide what your brain needs to clear toxic debris, rebuild damaged neurons, boost memory-related neurochemicals, and protect the mind's structure from age-related decline. This is not wellness fluff. We will explore the actual physiology, discussing mitochondria, neurotrophic factors, melatonin precursors, and neuroinflammation, citing research from journals like Alzheimer's and Dementia, Frontiers in Aging Neuroscience, and Neurology. After 60, several systems shift simultaneously. The brain's production of BDNF, the molecule that builds new neural connections and maintains existing neurons, begins to decline. The glymphatic system, responsible for clearing waste during deep sleep, slows down. The gut microbiome, communicating with the brain through the gut-brain axis, shifts in ways that reduce key neurochemical production. Mitochondria, the tiny power plants in each neuron, accumulate damage faster than the brain can repair them. What you eat before bed matters because the glymphatic system is most active during the first few hours of deep sleep. It flushes cerebral spinal fluid through spaces between brain cells, removing amyloid plaques, tau proteins, and other metabolic waste linked to Alzheimer's and cognitive decline. Eating foods that spike blood sugar, promote inflammation, or contain alcohol disrupts this system and hampers nightly repair. Providing your brain the right building blocks before sleep amplifies this process. The optimal window to deliver these neuroprotective compounds is 1 to 2 hours before sleep. 6 scientific insights guide this approach. First, aging brain mitochondria are central to memory repair, and one fruit compound targets them directly. Second, neuroinflammation, not aging itself, drives cognitive decline. Third, BDNF drops after 60, and specific anthocyanins can reverse this. Fourth, the serotonin-melatonin cascade during sleep is enhanced by one fruit's compounds. Fifth, certain fruit polyphenols reshape gut bacteria to produce brain-healing metabolites overnight. Sixth, deep slow-wave sleep is the single most undervalued tool for brain repair, and food can help access it. The first fruit is pomegranate. Its impact begins in the gut. Imagine brain cells as factories, each with thousands of mitochondria generating the energy needed for neurons to fire, consolidate memories, and think clearly. With age, some mitochondria malfunction, producing toxic byproducts. The brain has a quality control process called mitophagy, which removes damaged mitochondria. When this fails, damaged mitochondria accumulate, release inflammatory signals, and disrupt brain function, contributing to Alzheimer's and cognitive decline. Pomegranates contain polyphenolic compounds called L-gitannins and ellagic acid. Beneficial gut bacteria convert these into urolithin, one of the most potent natural activators of mitophagy discovered so far. A landmark study published in 2024 in the journal Alzheimer's and Dementia by researchers from the University of Copenhagen, in collaboration with the National Institute on Aging, used multiple Alzheimer's mouse models. They found that urolithin A improved learning and memory, reduced neuroinflammation, and actively restored the brain's mitophagy process and lysosomal function. The researchers discovered that urolithin A removes damaged mitochondria from the brain with an effectiveness comparable to NAD supplements, widely regarded as some of the most powerful neuroprotective compounds in modern longevity medicine. Consider this: a compound derived from a fruit produced naturally by your gut bacteria, performing at the level of one of the most expensive supplements on the market. A separate 2024 paper in the journal Nutrients confirmed that urolithin A activates the longevity gene, which is involved not only in mitophagy, but also in DNA repair, inflammation control, and cellular longevity across multiple organs, including the brain. Here's what happens inside your body when you consume pomegranate in the evening. The L-glutathione travel through the digestive system to the colon, where gut bacteria, particularly those in the Gordoniter and Helicobacter genera, convert them into urolithin. This process takes several hours. Eating pomegranate 1 to 2 hours before sleep ensures urolithin production peaks during the early hours of deep sleep, when the glymphatic system is most active and the brain is performing its deepest cellular housekeeping. You are essentially timing your food to match your brain's repair cycle. The practical protocol: consume roughly half to a whole pomegranate in the evening. If fresh pomegranate is unavailable, 4 to 6 ounces of pure pomegranate juice with no added sugar works. Pair it with a small amount of healthy fat, such as a few walnuts or a tablespoon of olive oil on a small salad, because polyphenols absorb better in the presence of fat. If your gut bacteria are imbalanced, supporting your microbiome for several weeks may be necessary for full urolithin conversion. Prebiotic fiber, fermented foods, and reducing ultra-processed foods support this process. The second fruit is blueberries. Beyond wellness hype, their effects on the aging brain are targeted and powerful. Blueberries contain anthocyanins, particularly cyanidin and delphinidin, which belong to flavonoids. Flavonoids can cross the blood-brain barrier, a protective membrane that filters what enters the brain. Once inside, they act in coordinated ways. A 2021 study published in Neurology by the American Academy of Neurology analyzed the diets of tens of thousands of men and women over many years. It found that flavonoids, especially anthocyanins in blueberries, blackberries, and cherries, were associated with a 24% reduction in cognitive decline risk. Even half a serving per day showed measurable benefits. Blueberry anthocyanins directly upregulate BDNF, brain-derived neurotrophic factor, which acts as the brain's fertilizer, promoting new neurons, strengthening existing connections, and protecting cells from death. Preclinical studies show blueberry supplementation elevates hippocampal BDNF, and the hippocampus is the brain region most responsible for memory formation and vulnerable to Alzheimer's damage. A 2023 clinical study in Frontiers in Psychology found that polyphenol-rich blueberry and grape extract improved episodic memory and cognitive function in older adults with mild cognitive impairment. Improvements were linked to increased BDNF and neuronal signaling proteins, including TREB, involved in consolidating short-term into long-term memories. A 6-month randomized control trial in 2022 in nutritional neuroscience found wild blueberry consumption improved information processing speed in older adults with mild cognitive decline. Research from the University of Cincinnati showed blueberry supplementation in middle-aged adults with insulin resistance and subjective cognitive decline improved memory interference and lexical access. Blueberries also appeared to enhance peripheral mitochondrial function, linking metabolic health, mitochondrial efficiency, and brain performance. Timing before bed matters. Memory consolidation occurs during sleep, especially REM and slow-wave cycles in early night hours. BDNF elevation from anthocyanins peaks roughly 2 hours after ingestion. Eating blueberries 90 minutes to 2 hours before sleep aligns peak BDNF and cerebral blood flow with the brain's active memory consolidation phase. The practical protocol: consume half to a full cup of fresh or frozen blueberries in the evening. Frozen berries are sometimes higher in antioxidants. Eat them plain, mix with plain Greek yogurt, or blend into a small smoothie with spinach and almond milk. Avoid sweetened products or added sugar, which spike glucose and disrupt sleep architecture. The goal is pure anthocyanin intake. The third fruit is kiwi, which begins its effect by improving sleep. The brain cannot repair effectively without deep, high-quality sleep, a principle grounded in neuroscience. The glymphatic system, your brain's waste clearance network, functions almost exclusively during slow-wave sleep. When deep sleep is insufficient or interrupted, your brain accumulates amyloid beta plaques and tau protein tangles, the compounds defining Alzheimer's disease at an accelerated rate. Sleep acts as the janitor shift for your brain, and if it doesn't happen, waste builds up. After 60, melatonin production drops significantly. By age 70, melatonin levels can be less than half of what they were in your 30s. This explains why many older adults struggle to fall asleep, stay asleep, or feel truly rested in the morning. When melatonin declines, slow-wave sleep becomes less efficient. Reducing the time spent in memory consolidation and glymphatic clearance phases, the brain's overnight repair cycle shortens. Kiwi fruit addresses this with remarkable precision. It contains natural melatonin, about 24 micrograms per gram, as well as serotonin, a direct biochemical precursor to melatonin. Kiwi is also rich in tryptophan, phenylalanine, and glutamic acid, amino acid precursors to key neurochemicals including dopamine, serotonin, and GABA. Essentially, kiwi is a neurochemical toolkit in a convenient, tasty fruit. A study in Frontiers in Nutrition in 2023 found that consuming kiwi with the evening meal significantly increased urinary concentrations of serotonin metabolite 5-AIAA and melatonin metabolite 6-sulfatoxy-melatonin. Compared to a control group, both fresh and freeze-dried kiwi improved mood, morning alertness, and reduced sleep disturbances. Researchers identified that kiwi's serotonin precursors, combined with the enzyme actinidin, which enhances amino acid absorption in the gut, create an earlier and more sustained peak of neurochemical precursors in the bloodstream. The brain converts these into melatonin and GABA, critical for initiating and maintaining deep sleep. A separate trial showed elite athletes who ate 2 medium kiwis 1 hour before bed experienced significant improvements in sleep quality and post-sleep recovery. A landmark study in the Asia-Pacific Journal of Clinical Nutrition found adults eating 2 kiwis 1 hour before bedtime had increased time in slow-wave sleep, the stage where the glymphatic system is most active, memory consolidation occurs, and brain cell repair is at its highest. The chain reaction is clear. Eating kiwi before bed raises serotonin, which converts to melatonin, signaling the circadian system to initiate deep sleep. Deep sleep activates the glymphatic system, clearing amyloid plaques and inflammatory debris. Simultaneously, the hippocampus consolidates daily memories into long-term storage. You wake up sharper, with a brain cleaned and reset overnight. The practical protocol: consume 2 medium green kiwis, skin on if tolerable, about 1 hour before bed. The skin contains 3 times more fiber and additional polyphenols. If texture is an issue, blend the whole kiwi into a small smoothie. Optionally, pair with a few tart cherries or a small glass of tart cherry juice for a synergistic effect on melatonin and sleep quality. Combining these 3 fruits strategically before sleep produces compounding benefits. Kiwi initiates the serotonin-melatonin cascade, deepening sleep architecture. Blueberries deliver anthocyanins that elevate BDNF as the brain enters its consolidation phase. Pomegranate feeds gut bacteria the L-glutathions needed to produce urolithin A overnight, stimulating mitophagy, clearing damaged mitochondria, and reducing neuroinflammation. After 60, compensatory mechanisms for suboptimal nutrition diminish. DDNF is lower, lymphatic efficiency reduced, gut microbiome diversity declines, and melatonin drops. Strategic, precisely timed nutritional intervention becomes essential. Regarding blood sugar, these fruits have low to moderate glycemic loads in the recommended portions. Blueberries have a glycemic index around 53, kiwi around 50, and pomegranate seeds roughly 18. For diabetics or highly insulin-resistant individuals, pairing with a small amount of protein or healthy fat, such as Greek yogurt, walnuts, or almond butter, helps stabilize insulin overnight, which is neuroprotective. An additional mention is tart cherries, especially Montmorency cherries, which contain melatonin and anthocyanins. A 2025 systematic review in Food Science and Nutrition found tart cherry consumption improved sleep duration, efficiency, and reduced inflammatory markers including CRP and IL-6. Preclinical studies showed tart cherry anthocyanins accumulate in aged rat brains, reducing hippocampal inflammation and improving working memory. While less accessible fresh, tart cherry juice can be included in an evening routine alongside or in rotation with the primary 3 fruits. Thanks for watching. See you in the next one.